Hello and welcome to our presentation on the separation of leaf pigments. This experiment investigated how light intensity and carbon dioxide availability influence the rate of photosynthesis in spinach leaves and how plant pigments separate by polarity during paper chromatography. Using a leaf disc assay, spinach leaves discs were exposed to different conditions: bright light with sodium bicarbonate, bright light without bicarbonate, low light with bicarbonate, and complete darkness. The percentage of floating discs was recorded over time as an indicator of oxygen production. Chromatographic separation revealed four major pigments: carotene, xanthophyll, chlorophyll A, and chlorophyll B, with RF values consistent with the relative polarities. These findings highlight the dependence of photosynthetic activity on light and carbon availability and demonstrate the chemical diversity of photosynthetic pigments. Chromatography is a fascinating lab technique that helps us break down mixtures into their individual parts. Take plants, for example. Their leaves are packed with a variety of pigments that work in harmony to soak up light energy for photosynthesis. While leaves might seem like they're just one shade of green, they actually house several pigments, including chlorophyll A, chlorophyll B, xanthophyll, and carotenoids. Each of these pigments has its own solubility and molecular structure, which means they move at different speeds when placed in a solvent on chromatography paper. By separating these pigments, we can see vibrant colors and gain a deeper insight into how plants harness and utilize energy. This experiment not only highlights the intricacies of photosynthesis, but also reveals that the colors we see in plants come from a collaboration of multiple pigments, all working together to sustain life. Here we have a graph showing data from a human-conducted experiment. This graph compares the RF values of several plant pigments separated by chromatography. We see that carotene has the highest RF value, meaning it's traveled the farthest and is the most nonpolar pigment. Chlorophyll A and B have progressively lower RF values, reflecting increasing polarity. Xanthophyllis have the lowest RF value, showing that they are the most polar and therefore interact more strongly with the paper. Overall, the graph clearly illustrates how pigment polarity influences chromatographic movement.
Methodology. The experiment consisted of two main procedures: a leaf disc photosynthesis assay and a paper chromatography analysis of spinach pigments. For the photosynthesis assay, uniform leaf discs, 6 to 8 millimeters, were punched from fresh spinach leaves, avoiding major veins, and vacuum infiltrated with either a 0.2% sodium solution or a distilled water containing a drop of dish soap to reduce surface tension. Discs were divided into four experimental conditions. The time for 50% of discs to flow was calculated for each replicate. For pigment separation, spinach leaf extracts were applied to chromatography paper with a pencil baseline and are developed in a solvent of 70% alcohol, and water. As the solvent ascended, distinct pigment bands were marked, dried, and their relative froth. Values calculated by dividing pigments travel distance by solvent front distance. The combined methodology enabled quantitative assessment of photosynthetic activity and qualitative identification of pigment polarity differences. In the graph, you can see the RF value from 0.3 to 0.9 and the replicate number from 1 to 3. Here are the results. Condition A produced the fastest rise in percent floating, confirming that both CO2 and light availability drive photosynthesis rate. Condition B showed slower rates due to CO2 limitations. Condition C displayed intermediate rates, while condition D showed minimal activity. Chromatography results revealed pigment separation consistent with polarity. This demonstrates the diversity of leaf pigments and their differing roles in light absorption. Results showed that discs in the bright light with bicarbonate condition reached 50% floating, the fastest, confirming that both light and CO2 are essential for efficient photosynthesis. Low light and CO2 limited conditions produce slower rates, while darkness resulted into photosynthesis activity.
And so we start with the conclusion or expected outcomes. It is expected that spinach leaf discs exposed to bright light and sodium bicarbonate will show the highest photosynthetic rates, demonstrated by rapid oxygen production and shorter ET50 values. Conditions with limited carbon dioxide or reduced light intensity should result in slower floating times, while those kept in darkness will exhibit minimal or no photosynthetic activity. These findings would support the hypothesis that both adequate light energy and a carbon source are essential for efficient photosynthesis. In the chromatography portion, distinct pigment bands, B-carotene, xanthophyll, chlorophyll A, and chlorophyll B are anticipated to separate according to polarity. With B-carotene traveling the farthest and chlorophyll B the least, together these results are expected to confirm the interdependence of light and carbon dioxide, CO2, in driving photosynthesis, and illustrate how pigment diversity contributes to capturing a broad spectrum of light for energy conversion. And as we could see here in the comparison, this is our comparison between both of our experiments, the other one being the AI and our original one. As we can see, there's a big difference in terms of the chlorophylls and antholysis, the chlorophyll and the serotonin. We see that the orange for many of them is higher except for xanthophyll. And yeah, further research ideas and or acknowledgements, further future studies could investigate how various leaf types impact the results of pigment separation. For instance, examining spinach, kale, romaine, or red cabbage might reveal variations in the types and quantities of pigments, solvent used in the chromatography process to determine how the polarity of the solvent affects the distance each pigment travels. Additionally, students could analyze different types of chromatography paper, the amounts of pigments applied, or the levels of solvent to gain a deeper understanding of how these experimental factors influence pigment movements and separations. And our references are down below. As we can see, we have ChatGPT, we have LibreText, we have LibreText Bio, and we have Whole Science as based on our other experiment. And yeah.